right, thanks for the introduction. Sorry, excuse me. Okay, so today I'm going to be talking to you about BIOSCAN-5M, 5 million, which is a new data set that we've released. Came out recently last year and was presented at the NeurIPS conference in the Datasets and Benchmarks track. So it's a large data set that can be used for a variety of different applications, so different kinds of things you could benchmark with it. So I'm going to go over the motivation for the data set, why it's of interest, and some properties about it. So first of all, why do we care about biodiversity? Well, biodiversity is incredibly important to human life and the life of the planet in general. But despite this, Earth's biodiversity has been decreasing significantly over the last few decades. So this graphic here from the WWF Living Planet report, produced by Visual Capitalist graphic. And so we can see that there's a variety of things which have been impacting biodiversity in increasing order of impact: climate change, pollution, invasive species and disease, species overexploitation, and changes in land use and sea use. And all of these things are human-caused factors. And on aggregate, there's been a 68% decrease in vertebrate species populations between 1970 and 2016, which is an enormous decrease. But that is vertebrates in general. Vertebrates includes all sorts of animals, not just insects. So what about insects? Well, here's a recent profile from New York Times Magazine from a few years ago. Arthropods and other insects are being lost even more rapidly than the average for vertebrates. And so we have a decrease in 75% in insect biomass across a variety of research places in Germany between 1989 and 2016. So on a shorter period of time, there's been a larger reduction in insects compared to vertebrates overall. And other kinds of vertebrates, say birds and whales, they're more charismatic, they get more attention. But insects are pretty key to a whole bunch of things that we really need. Like they act as pollinators for agriculture. We'd have enormous agricultural collapse if we don't have any insects. And maintaining biodiversity is important for maintaining the variety of species that we have and a healthy ecosystem. So in this project, we're trying to help map insect biodiversity around the world. And this is part of a group called the International Barcode of Life, IBOL for short. And within that, we're the BioScan team, and we're working with the Barcode of Life data system, BOLD. A key place for this is the Centre for Biodiversity Genomics, which is based in Guelph in Ontario, Canada. And so there's data that's collected from all around the world, and it's sent over to the Centre for Biodiversity Genomics, and they do DNA barcoding on it, which I'll explain in a moment. The group is over 1,000 over 1,000 researchers in over 30 countries. So it's really a worldwide effort. The data collection, we have traps that you can see in the top left image, traps that are placed in the wild, and they collect insect samples for a given location. The insects are imaged with very high resolution microscopes, and then they're sequenced with DNA sequencing techniques to extract the DNA barcode, and then they're housed for long-term storage and preserved with ethanol. So the DNA barcoding, that's something that I think a lot of people won't be familiar with. You've heard of DNA, of course. DNA barcoding specifically is sequencing a particular part of the DNA. So rather than spending a lot of effort sequencing the entire genome for all of these insects, which isn't scalable, instead the entomologists have worked out a particular component of the genome. It's actually specifically part of the mitochondrial DNA rather than the animal's main cell DNA. And so this particular gene, the cytochrome C oxidase 1 gene in the mitochondria, this is very indicative of which species specimen is. And it's used across all vertebrates, not just insects. So by sequencing this particular gene, we're able to ascertain which species it is very reliably. So a given barcode will only occur for one species. You don't get the same barcode across multiple species. And within one species, there'll only be a handful of barcodes that appear because they're highly conserved. So in a sense, a barcode, a specific barcode is an indicator of like a subspecies. And they get grouped together into barcode index numbers, bins, with like an automated clustering process that the Barcode of Life database uses. So that gives like an indication of the subspecies. So there's many millions of sequences on BOLD, I think over 20 million now. And of these, there's 5 million insects, which is the data set that we've released here. So as an example, with the high resolution images, they're using a Kayent digital microscope. I can't remember what zoom level it is, but the insect is only about 2 millimeters across. So it has to be a very high resolution image. We end up with like a 1000 pixel image at the end. The data set for our 5 million specimens. So we've got a variety of different characteristics for each specimen. So we have the DNA barcode that we've just been describing. And note that, as I said before, this is a single gene. So this is only 660 base pairs long. And it's just made up of the letters C, G, T, and A, plus sometimes special character N that indicates there's a missing bit of DNA. So there's only 5 characters. So it's quite short, 660 base pairs long. And we've got our high resolution image shown in the middle here. There's a taxonomic classification that's done by an expert who looks through at the samples and determines a label. For this data set, we actually got the family level automatically generated by a pre-existing model. So the vast majority of specimens are labeled to family level because this is automated or highly automated. Whereas the things that are more detailed than that, a human has had to look through the specimens to give the label. And this can take quite a long amount of time for them to do, depending on what it is. Sometimes it'll take, short amount of time because it's easy to determine the species, but sometimes there'll be species that look very similar and are very hard to tell from the image. Sometimes it'll be impossible for the expert to tell from the image. They'll have to get the specimen out and examine it, and that could take a couple of hours. They may even have to dissect it. And also, there's a very long tail for the distribution of species. So that means that there's species that have never been observed before. And we can observe them in this data set. So we have examples of new species that haven't been named. And for those, it takes even longer to determine that it's a new species rather than existing species. The barcodes get clustered into bins. So we have the bin ID here, and this acts as like a subspecies level label. And this is automated. So although we don't have species labels for everything, we have the barcodes for everything. We have the bins for everything. And the Center for Biodiversity Genomics actually argues that we should just abandon the idea of using taxonomic classification, which isn't based on evolution. It's just based on like a hierarchical organization that people have used for the last 150 or so years. So it's just a slowly evolved human system rather than something that maps natural evolution. So they argue that we should use the bins instead because they're objective and are not subject to human error as much. And we also have annotations about the size of the insect because we have the magnification factor for the image that tells you the scale of the image, and using a mask we determine how large the insect is. But we don't have the biomass; we just have the size of the insect. And we have the location it was collected at. So a variety of different types of data available. And here's some more samples of what the insects look like. So they cover a whole bunch of different categories of insects that you'd be familiar with or some that you won't be familiar with. So we have moths, butterflies, ants, bees, a heck of a lot of flies, various different types of flies. And the data has been collected from all around the world, as I said before. So you can see a map of very diverse coverage here, with the largest amount being collected from Costa Rica, because Costa Rica has got a particularly large amount of biodiversity, and there's been a lot of effort to map that. So with regards to the statistics of the data set, we've got this release, BIOSCAN-5M, which is an upgraded version of our previous version, BIOSCAN-1M. It's 5 times the size. And we have 5 times as much data. And so you can see the coverage, as I mentioned before, we have basically everything labeled to order level, and nearly everything to family level, 96% to family level. But then when you want to look at the more fine-grained classes, but genus level and species level, we only have 24% and 10%. But bearing in mind this is a lot of data, a quarter of our data being labeled to genus level is still over 1 million samples, and it's larger than the previous release was in its entirety. So it's a very large amount of data. And everything's labeled with the DNA barcodes, as I mentioned before. So the fact that it's paired data enables a lot of interesting things that you can do. You have a taxonomic label, least to order level, a DNA barcode, an image, and a location for the specimen. So there's a lot of diversity in sort of things you can do with this. We've also taken some care to clean up the data a bit compared to what we got from BOLD. So there's a couple of ways in which there's redundancy in the labels for the data. So the species name, I'll just show an example here. The species name is the first word of the species name is the genus name. This should be always consistent. So we can check whether this is the case, and where it isn't, flag it up as something that needs to be checked. We also fix some typos. This doesn't typically occur with the things that have proper scientifically established names, because those are kind of selected from a drop-down menu, I think. But the species that haven't been observed by science before have placeholder names. So here's examples of things with placeholder names. You can see the species name is the name of the person that found it, and then some number, or bold for the dataset name, an identifier, or Malays for Malays trap. So there's some different ways of naming the placeholder names you can see here. So we have found some cases where the placeholder name was done inconsistently. So 2 samples that should be the same placeholder species would be named with different versions of the placeholder name. So we changed that so they collapsed and collided with each other, so that they're not identified as different species when they're the same novel species. And then the other thing we did is, as I said before, the DNA barcodes are very indicative of which species it is. You don't get the same barcode across different species. So we checked if that ever occurred, and if it did, then we flagged it as a conflicted label. So that means that there's either disagreement between annotators, or something's being annotated incorrectly. It could be an incorrect match between the image and the label somehow. So something like that's gone wrong. So we flag these things up. So in this regard, we've done the best that we can to clean up the data set. There's also an image-based filter where we rejected samples that were incorrect image, so a partial image of the insect, or an incomplete image of the insect, or the insect wasn't in the well for the imaging, something like this. So as we have the data where we have proper species names, so scientific species names here, here's some examples. And then we have specimens that are being observed by science for the first time, at least, which have placeholder names. This then poses an opportunity to do an interesting task with the data set. So rather than just trying to label all of the species names, given the data set that we have, we want to be able to address the task that's actually experienced by annotators as they're doing this. So the whole goal of using this data set for biodiversity monitoring is we want to try and automate this process that's really slow, which is trying to annotate the images. And so the best way that we can do that is not just by using the names from the categories we already have, but also being able to identify if this is a new species or grouping together samples from species that haven't been observed together and saying they go together. So in order to facilitate that, we set up the partitioning for the data set so that there's a seen partition and an unseen partition. So the seen partition is made up of all of the species names that are proper names, scientific names, species that have been seen before, and the unseen samples, the unseen partition, are placeholder names that are indicative of what we'd experience for things that have been observed for the first time, because these ones were observed for the first time when they were recorded. And they will, go through the process and they'll get proper scientific names after a while, but they don't have proper scientific names yet. So this means we can put these two tasks, both a closed world task, trying to identify names of seen species, and an open world task, which is trying to group together examples from unseen species. To make this a bit more standardized, for the unseen species, we only included species for which the genera, the genus, appears in the seen partition. And so for things that don't fall into that category or which there's too few samples to make a test partition, we put those in the held out set. The held out's just sort of everything else. Within the seen partition, we make a training set you can train the model on, in addition to all the other stuff that we don't have species labels for, which you can use for pre-training, validation partition, and a test partition. And then for the unseen data, we make similar partitions, except instead of being training, this is reference keys that you can use for reference queries, reference lookups. And then you query that with the validation and the test partition. That's the idea. And there's a very long tail for this distribution, as I said before. So we flatten that by trying to take around 25 samples per test. This sort of increases linearly if there's below 25. And if there's hardly any, then we just don't put anything in tests. The val partition is not done the same way as the test. The val partition, it just has the same distribution as the training. So we have the training distribution, which is the majority of data. Validation, the same distribution as training, and test is a flattened distribution to try and make it more about the identifying all the classes rather than the majority class. So we end up with these partitions. The vast majority of the data doesn't have species labels, so that becomes pre-training. We can use this for self and semi-supervised training. And there's 4.5 million samples here. Although every specimen has a barcode, not all the barcodes are different, of course. So on average, there's about 2 images per barcode. So if you're interested in just using the barcodes, then you'd have half as many records to use because you have to remove the redundancy in the barcodes. Then the scene partition for the closed world task, the training partition, validation and test partitions. And then the open world, that's on the unseen, you have the key, val, and test similarly. And then finally, the held out data, which you could use for, say, training a novelty detector. But at the moment, it's not evaluated on. And there's a tree diagram. You can see the majority of the data in blue is pre-training, and the vast majority of that is labeled to family level. And the green is the seen data. Red is the unseen data. The unseen data is, of course, smaller than the train because the majority of species you observe are going to be seen before. So an important note about this partitioning system that we used is that it does mean that there's a different class balance for the different partitions. So pre-training, which is basically the distribution we have for the data overall, 75% nearly of the data are flies. And then after that, we have some other insect species in blue. And then we've got spiders and cholera. If we look at the scene data, the distribution there is a bit flatter. So the flies don't dominate as much, but they are still the majority class. This is the family level, sorry, the order level that we're looking here. So there'd be a whole bunch of different fly species within this, very large number of fly species. Turns out there's a lot of flies. And then in the unseen data for the open world task, the distribution is actually quite different. Here we have an almost equal split between the flies, the ants slash bees, these are in the same order, and the moths and butterflies and things like that. And there's not very much of the other data. And the reason why this is happening is because the species that are being observed for the first time are coming from species that have either not been studied as much in the past by science, or they're coming from orders where there's a higher speciation rate. So the new species occur more frequently. So these kind of factors are the reason why we have this different distribution. So things that have been studied before look like this, things that have not been studied as much look like this. That's sort of the gist. Oh, and the held out data, where we have the unseen genuses or genera, actually that's dominated by the moths more. So at the genus level, there's a lot of new moths and more ants than flies as well. So to summarize for BIOSCAN-5M, we've got 5 more samples than we had for BIOSCAN-1M. A lot broader coverage. So we've gone from 3 to 47 countries the data is collected in. A much greater diversity of the data. So we've gone from only 1 class to 10 classes and 16 to 55 orders. The data is being cleaned, which should improve the quality. We've added in the geographic locations and specimen size information. And we did the partitioning so you could do closed world and open world tasks. And we provided some benchmarks on both of those tasks using different versions of the modalities. And if you want to use the data set, it's pretty easy to get started. You can just pip and install the BioScan dataset package, and you'll be on your way straight away to automatically download the dataset. You just need like 3 lines of code to get running. And just quickly go over some examples of the work we've done with this. So these are experiments that we had in the paper. These are building on previous work that we've done and applying it to the new data set. So we did some work on zero-shot clustering. So here the idea is you take a pre-trained encoder that's been trained on some data. In this case, we used ImageNet pre-trained encoders and deploy it on some new data for inference. And the new data has never been seen by the training the training of the model. We freeze the encoder and we reduce the images with UMAP with 50 dimensions for the output, which is a lot more than people usually use for UMAP. Usually people just use 2 dimensions for visualization. But if you do it with larger amount, it doesn't destroy the data, and it preserves the clustering information in the reduction. And that sets it up very nicely for using a clusterer afterwards to identify the clusters that UMAP has preserved. In this case, we used agglomerative clustering for that. And when we do this, we can see that we have, here we're measuring the agreement between the clusters that we created from the embeddings versus the ground truth labels. We did this on the test data, and we find that the image information, so if you try and embed the images, you can get good performance, like 90% with Dino, if you're looking at very coarse level. But once you go to past order, this drops quite precipitously down to only around 20% with the best model Dino when you're looking at species level. And so like 65 for family and 37 for genus. If you compare that to using the DNA barcodes, the performance is very high and stays very high the whole time. And the reason for this is the barcodes are the method for identifying the species. It's the whole point of the barcode. They picked out the 660 base pairs that indicate which species it is. So we don't need to do much work from that. So if you have got a reasonable encoder, then you can get clustering that agrees with the labels, the taxonomic labels, quite readily. This plot is the DNA barcode experiment here is cheating a little bit though, because as I said, there is redundancy in the barcodes. The barcodes occur about twice on average each, whereas images are all unique. So clustering things that are repeated makes things easier, of course. So if we deduplicate the records and just look at every barcode once, so we dropped like half the images, and the barcodes are dropped to be unique, then we can see the performance is still actually pretty high for the clustering on the barcodes, but not as high as it was before. But we still get 75% agreement with the species. So this indicates that there's just without any training on the data, you can get very far with the barcodes. But the real task is how do we handle the images? Because that's the thing that's cheaper to collect. So really, we'd like a method that you can use the images for and get good labels. The other experiment was using transformers on the barcodes, and here we pre-train on the pre-training data, of course, and then we fine-tune on the training data. And the training here was using the BERT methodology with masked language modeling. And we mask out particular tokens after creating our tokens using k-mers. So in this example, a 3-mer. So we group together 3 consecutive tokens, 3 consecutive letters, sorry, in the genome, and that becomes 1 token. Anytime there's a missing character with an N, that becomes an unknown token. Just to keep the space in the dictionary lower. And then we mask out some samples. We ask the model to complete the masked out elements in the input, and that's the pre-training. Then we fine tune by taking the average of the embeddings for the representations and train that on the training data. And then we find, with either the linear probe on the pre-trained model without training, we get very high performance, like 99%. And if we do the fine-tuning, then we get 99.7%. But we could do that with any pre-training data, any of these pre-trained models that's not even trained on the barcode data, just trained on human DNA. So the training step, the fine-tuning, makes it really easy to get very high percentage accuracy. But that's because the barcodes are so consistent. So these aren't held out barcodes, but within seen species. If we look at trying to handle the unseen species, so this is here we're doing genus level accuracy on unseen species, the performance is 78%. Oops, 78%. So it's still good, but this is where there's the challenge. How to handle the unseen data, unseen species with the barcodes is still not solved. And the third experiment, we're doing a clip-style model, and we're doing multimodal training. So we're aligning the embeddings of an image encoder, DNA encoder, and text encoder, and creating a shared embedding space for the 3 modalities. And the utility of this is after we train the model, we can not just use it on the seen species, but we can also use it on the unseen species by creating a data bank of keys that we can have on record for unseen species that have been, say, observed for the first time. So after someone sees a new species, they put it in the data set, and then we can do queries with that one in the future. We don't have to retrain the model every time a new species is discovered. And so for this, we find trying to map the DNA of a new specimen to DNA that we have in the query bank. that works incredibly well. We get very high accuracy. So here everything's matched to species level. If we match images to images, then this works generally quite well, but not always as accurately. So in some cases, only to family level in examples. And trying to map image to DNA, which is cross-modal, is generally high, but it's a bit tricky to see the overall results there. But in the results, we see that mapping within modalities, so image to image, works more accurately and than trying to map from image to DNA. So this remains an open problem for this setting, trying to align these modalities more carefully so that they are aligned even on unseen data. Okay, so to conclude, this has been introduction to BIOSCAN-5M, which is a comprehensive multimodal data set for biodiversity research. We've got 5 million specimens. There's a lot of modalities, images, DNA barcodes, taxonomic labels, geolocation, size information. We've got some tasks that we demonstrated, but you can see the kind of things you could do with this data set. The question of how to handle the barcodes on seen samples is basically solved, but how to handle them on unseen specimens is tricky, and trying to deal with the images is also still tricky because the images are a little harder to identify the species from. I've got a whole bunch of collaborators. Thank you to all of them for collaborating, and I'll close there for questions.
Cool. So we do have a couple of questions. The first one is, do your DNA barcodes provide insights into evolutionary branches?
Do the DNA barcodes divide them into evolutionary branches? For the question?
Yeah.
In a sense, the distance between barcodes increases with taxonomic distance, but they're not phenotypic, so they don't indicate any properties of the specimen, how it looks. So yes, just because of the nature of evolution pushing these things away over time. Yeah.
A quick question, which is just clarification on the data set. What is meant by keys in the training column?
The idea is you would have some records that you've cached beforehand, and then you can do queries against that data bank that's there to see which key you match up with. So it's the retrieval setting, which is common with CLIP. Is that clear?
I think it'll work. Next question is, how do you guarantee the samples of the training set get into the test set, given the wide variety of data where different samples of the same classes could be there?
Well, we partitioned the data so that was the case. So that there was a complicated partitioning process. So we made sure that for every species that had enough samples and more than one barcode, it would have at least one barcode in the test partition, at least one in the training partition, and try to get a comparable number in the training partition, in the test partition across the different species. This is like a multi-dimensional partitioning that makes it non-trivial, but managed it pretty well. So
And then we'll take one last final question here, which is kind of a two-in-one, but it's around the issue of how do you assimilate and collate photos from different angles? For example, you might have an insect where it has their wings spread, it doesn't, it's from different angles. How do you address those issues in your data set?
We only have one image per specimen, so we're not going to have an image of the same specimen with its wings in different positions. With regards to how do you do diversity across different poses of different specimens, it's a challenge. That the insects are supposed to be arranged in a relatively consistent way when they're placed in the dish, but it's not always the case.
Hey, how's it going, everybody? So real quick before we get started on the thing I'm here to talk about, I did drop a link to a blog post. In this blog post, I talk about the BIOSCAN-5M dataset. I talked to Scott actually when we were at NeurIPS. And so I've parsed the data set, small sample of it, 30K, and done some kind of embeddings computed for that and all that stuff. So you can, play around with it, the data set, and kind of check it out and, explore it more deeply if you're into this data set and into the presentation. Really good data set. You can also view it on map. However, I forgot to pass my Mapbox token. In the blog, I'll show you how to do that. But if you want to explore this, get hands-on with it, I've dropped a link to a blog post there. Jimmy, I'm sure you'll resurface it and drop that back as well.